Okay, so yeah, next talk. So this will take us back to a little bit more technical information. So just you know, we've sort of set the stage for this with the last couple of talks, but it's a you know, we always find it useful to give everybody at least a you know, an introduction to how, you know, specifically how we use mass spectrometers and mass spec data to identify peptides and proteins. So this is where we start out. So you know, I talked about this at in the last talk. We always start with a trypsin digestion. So we can start with, you know, in this case, we're looking at a protein in gel, but it can be any protein sample. Usually these are complex mixtures for most of the projects we work on. So we do that first step with digest the protein or the protein mixture, and then we do some combination of either, you know, one-dimensional or two-dimensional HPLC separation. We may do the TMT isotopic labeling, but basically you know, this process through which we identify peptides and then proteins is essentially the same regardless. So we, you know, these two photos here are of older LC and mass spec instruments that we have, but they, you know, they all have about the same configuration. So we inject this tryptic peptide mixture into the LC-MS system, and then where things get interesting after we've separated out the peptide mixture by reverse phase chromatography is this electrospray ionization, which Rick talked about. So this is a similar diagram to the one he showed. And so here, this is the, you know, the end of the HPLC column, and so this is where you get the gas phase ionization, and you begin the mass spec analysis. And there are two, you know, leaving out tandem mass tag and MS3 for the time being. We do you know, two different measurements here: a MS1 scan to detect intact peptides, and then we do the MS scan, the tandem mass spec scan to get peptide fragment information, and this is how we can identify the sequence of the protein. So we'll just look in a little more detail using an example of how that happens. So if we do a tryptic digestion of bovine serum albumin, this is the result of that. So over here on the left, that's the sequence of the amino acid sequence of BSA, and it shows you in alternating black and red and uppercase and lowercase the tryptic peptide products. So trypsin cleaves on the C-terminal side of the basic residues, lysine and arginine. So you get this pattern of peptides anywhere from a single amino acid of 146 Daltons up to a, you know, 2400 Dalton peptide. So there are some, you know, potential problems here in terms of getting perfect mass spec data and identifying and quantifying everything. Mostly related to, you know, gaps in sequence coverage. So, you know, with really small or really large peptides, you're not going to be able to see everything. So not everything's going to be efficiently ionized. Not everything is going to be detectable and distinguishable in sequence terms from any other possible protein that could contribute a peptide. So you're going to inevitably end up with sequence coverage gaps. So this is almost universally true of proteomic analysis. You don't get perfect data. It's not like you know, Edmund sequencing where you start at the beginning of the protein and you sequence all the way through. And you know, this is, you know, the reason it's called shotgun proteomics is because it's hit or miss in that way. You just blast the protein apart with a protease and then with a mass spectrometer, and you gather information on what you can still see, and if you can get enough information to distinguish one peptide from any other possibility, then you can get usable data. So you don't need perfect sequence information to identify and quantify proteins, which is good. So continuing with our albumin example here, if we load 20 femtomoles of trypsin digested BSA on our LC-MS system, we get a chromatogram that looks something like this. So you know, in this case, we've run a 30-minute acetonitrile gradient starting from essentially just aqueous up to 35 to 40% acetonitrile, we get separation of the peptides in these peaks that you can see from about 5 minutes out to 15 or 20 minutes. And then at the end, that's just a wash that rinses off noise and undigested protein or whatever else is in there. But what we're interested in is these peptide peaks over the first 20 minutes of the gradient here. So most tryptic peptides will elute in that range of acetonitrile concentrations. Then we want to do our two mass scans. Then first we want to look at intact peptides. So this is traditional data dependent analysis as opposed to the data independent that I talked about in the last talk. So here we've got to be able to detect a peptide before we do anything with it. So we're starting out just scanning across a fairly broad M over Z range from 375 out to 1500. So most tryptic peptides will have a charge state that will fall within that range. So this is what we see at about 15 minutes into the gradient. So this RT here, that's the retention time. So 15 minutes in, we're seeing this collection of peptides elute, or at least we hope they're peptides. So in data dependent acquisition, once we've got that MS scan, we want to try to isolate in sequence, starting usually with the most intense, each of those peptides and fragment it to get sequence information. So we do this by isolating, each of the peptides in the linear ion trap that Rick talked about, and using it as a collision cell to fragment each of those, isolate and fragment apart each of those peptides. So then we get an MS2 scan. So the MS scan, this is post-fragmentation. So in this case, it's MS2 728.66. So if we go back to the last slide, we've gotten through to, I think, the 7th or 8th most intense peptide that was detected in the MS scan. So here we're isolating this one here at 728.66 and fragmenting it apart to get this pattern of fragment ions. This is what we're going to use to reconstruct the sequence of the peptide. So we need to look exactly at how that fragmentation is happening in order to understand how we get the sequence information. So most of the time we're doing collision-induced dissociation or high-energy collisional dissociation, and those techniques tend to break peptides apart at the peptide bonds. So you generate a series of B ions and Y ions depending on which end of the peptide holds on to the charge, the N terminus or the C terminus. So these brackets here showing B1, Y2, and so on. That shows you how the peptide fragments. There are other possibilities with electron transfer dissociation that break the peptide backbone at different places, but to give you the same kind of sequence revealing information. So in the example, we'll stick with CID. So we're generating B and Y ions. So to look at a specific peptide, then this is the result of that process. So we've isolated this peptide sequence here at the top and fragmented it by CID. And these are all of the possible B and Y ion products of that fragmentation that you can see. So each peptide is broken in once. It fragments, each molecule fragments in one place. And for the purposes of this talk, we'll just say that's random. So if you've got enough molecules, which you generally do in mass spectrometry, you get all of these, in principle, all of these different B ions, depending on where the fragmentation occurs. So ion B1 is just the N-terminal tyrosine amino acid here, but B2 is the first two amino acids from the N-terminus and so forth. And then the Y ion series reads from the opposite end of the peptide. So you can start to see that this is going to generate mass ladders, which will correspond to the amino acid sequence. So you can determine what amino acid is present at any point in the sequence if you have the two B ion fragments or Y ion fragments on either side of that amino acid position by just subtracting the mass difference, the M over Z. So this is the then the annotated MS-MS spectrum from the fragmentation of that same peptide we were looking at on the last slide. So you can see you've got the overlapping B and Y ion series, and you've in the old days you would do this manually, and just by trial and error work out this ion mass minus this ion mass corresponds to the amino acid, the mass of the amino acid aspartate or whatever it is. So this is how you read out a peptide sequence from MS-MS data. Now, of course, to do this to identify 10,000 proteins in a cell lysate with just a few hours of mass spec time, this has all got to be automated because we're talking about tens of thousands of spectra being generated per hour of instrument time. And there are other problems with data analysis involving presence of noise and non-ideal fragmentation patterns and things like that. But at least the first stage of data analysis is based on database searching, which is comparing libraries of theoretical spectra to experimental data and determining how well they match. And this is possible because we've got genome information on most of the species we're studying here. So, if you're looking at human proteins or mouse proteins, the sequences of that can be translated out of all of the genes in the genome. So it's possible to actually take the genome, translate it to a proteome, and do a theoretical trypsin digestion and a theoretical tandem mass fragmentation for the entire proteome and generate a library which should contain a theoretical spectrum corresponding to any peptide you might observe in mass spec data acquisition of a sample derived from that species. Now, of course, that doesn't work perfectly, but it works well enough that you can get some pretty good data. So there are search engine algorithms that are designed to perform this comparison of the experimental spectra to the theoretical spectral library. So basically, in simple terms, you just look for masses that should be there, and your score goes up if they are there. If there are masses present that shouldn't be there, the score goes down. That's wildly inappropriate oversimplification, I'm sure, but it gets the point across, and that we'll get it in data analysis talks later. We can you'll probably get a better idea of some of the details of how that works. But basically, you set a significance threshold, and you generate a list of peptides that you identify based on that database search, and then you compile everything at the end. And then with our example, 20 femtomoles of albumin, we've identified and confirmed the peptides that are highlighted in yellow from that sequence. And again, we have, of course, we have sequence gaps, but there's more than enough of the protein sequence identified to distinguish it from any other entry in the database, which is what we need. So then we'll wrap up with just a little bit about applying this to identification of PTMs. And this works essentially the same way. So you can probably see that as long as you know the mass of the PTM and where you expect it to be. So say we're talking about phosphorylation here, we know it should be, if there's a phosphorylation present, it's going to be on serine, threonine, or tyrosine, and we know what the mass change is going to be. So we can just build that into the model. We just tell the database search algorithm to look for that mass modification at every time it sees a serine, tyrosine, threonine. But the problems are more related to the sample, to the problem we're trying to solve. So, there are a couple of issues. One is stoichiometry. So, continuing with the example of phosphorylation, it's not all or nothing. You can turn on a signaling pathway by phosphorylating maybe 5 or 10% of a protein in some cases. So you're fighting that. not all of the protein has the modification you want to see. The other, another issue, skipping down to the third one, is stability. So we'll look at a phosphorylation spectrum on the next slide and see what the problem is there. If you lose the modification, then it's harder to find. Again, it's the same problem as on number one here. Not every peptide you detect has the modification you want to see. And then the sequence coverage gap sometimes is really what gets you. So if all you're trying to do is identify the protein, and you can only see 40% of the sequence, that's more than enough. But if you're looking for a PTM, you have to have the peptide that contains that PTM. And if it's not a good tryptic peptide, then you're just going to be blind to it. So you can try to work around that by using alternate proteases and doing theoretical digests to come up with a good strategy like we were talking about earlier. So we'll just wrap up with the quote unquote typical phosphopeptide spectrum. And so this is what happens when you take a phosphorylated peptide and fragment it you do the CID fragmentation. By far the most abundant product of that fragmentation is the neutral loss of the phosphorylation, which gives you a nice signature spectrum. So it's easy to look at this and say, hey, that's a phosphopeptide, but it makes it a lot harder to get at the BNY ion sequence information that tells you what that peptide is. So this kind of illustrates the challenge of applying mass spectrometry and proteomics to looking for post-translational modifications. And this is why you almost always have to enrich for the protein you're interested in or the modification you're interested in. So we'll wrap up there, and